Hello and welcome to the Sound of Science. Today I want to speak about the differences between the mRNA vaccines developed by Pfizer, BioNTech and Moderna to the vaccines developed by Oxford Astrazeneca collaboration. We'll talk about the main differences between them. We will begin with the Pfizer, BioNTech and Moderna vaccines. So to understand these vaccines, we need to understand the coronavirus. As you can see, this is the scheme of the virus. It has the spike proteins, the famous proteins towards which all the vaccines are developed. And within that viral particle, we can see the RNA, which is the genomic content of the virus. Following entry of the virus, there is an interaction between the spike protein of the virus and the receptor expressed on epithelial cells of the upper respiratory system. This receptor is called ACE1, as you can see here on the screen. And following interaction or successful interaction between the spike protein and the ACE2 receptor, there is an entry of the viral particle into the cell and therefore infection. So typically what happens in the cell is that DNA within the nucleus of the cell is being transcribed into RNA. This RNA molecule will then export into the cytoplasm, and then it will be translated into proteins using the cell's ribosomal machinery. What happens when a coronavirus infects the cell? It actually hijacks the cell's systems that take RNA and translate into proteins. So the coronavirus actually doesn't go into the nucleus, it doesn't spread its genetic material in the nucleus, it simply spreads it in the cytoplasm, and the cell itself translates the RNA into a protein. So to generate the vaccine, what the researchers in Pfizer, BioNTech and Moderna did was to take from the entire RNA genome of the virus, they took the segment that encodes to the spike protein. So this segment alone was used to synthesize a synthetic version of this RNA segment. So the vaccine doesn't include a viral RNA, it includes a synthetic RNA version. And in addition, since RNA is a highly unstable molecule, this synthetic RNA was modified in order to increase its stability such that it will remain for at least 48 hours in the body. Another thing is that this RNA molecule, the synthetic RNA molecule, was encapsulated within a lipid particle called a liposome. And this complex of the liposome that includes the RNA molecule inside it is injected into the muscle. And following injection, roughly 2 weeks following this, there is a production of antibodies by the immune system that is characterized by IgM antibodies followed by production of IgG antibodies. As you can see, the levels of these antibodies decrease with time, and this decrease is different between each individual. Let's move now to the vaccine developed by Oxford Astrazeneca collaboration. This vaccine is based on an adenovirus that was isolated from chimpanzees. That's why it's called the chimpanzee adenovirus 1. And the reason for the development of this particular adenovirus is that it typically persists in chimpanzee populations, and therefore human population was not exposed to it, and therefore will probably generate a strong immune response towards it. So this is the rationale for using a chimpanzee adenovirus. Now, since adenoviruses are known to cause various illnesses, including the common cold, the researchers had to take out genes that are relevant to the virulence of this virus, as well as genes that are involved with replication, because we don't want this particular virus to replicate inside our bodies. What did the researchers insert inside the virus? So you remember that the genome of the virus, of the coronavirus, is an RNA form. So they had to change this RNA into DNA, and then they took the DNA particularly or specifically of the spike protein and insert this piece of DNA that encodes the spike protein into the genome of the adenovirus. Okay, so the genome of the adenovirus is DNA in contrast with the genome of the coronavirus, which is made of RNA. So now we have an adenovirus that cannot replicate inside our cells, but it can deliver the DNA for the spike protein. And that's exactly what it does. It can deliver its DNA into the nucleus of the cells. Within the nucleus, this DNA will be transcribed into RNA, which, as we know, will be exported then to the cytoplasm and be translated into a protein. It is important to understand that the DNA of the adenovirus doesn't incorporate into the genome of the host cell of our cells. Therefore, following injection of this particular adenovirus or the vaccine into the muscle of the person that is being vaccinated, there is once again production of antibodies roughly 2 weeks after injection, again of both IgM and IgG. And so in this short video I wanted to explain the main differences between the vaccine developed by Oxford Astrazeneca and the mRNA vaccine developed by Pfizer, BioNTech and Moderna. Until the next video, stay safe and thank you.